Good day everyone, we are Group 8 but before anything else, let me introduce myself first. I am Jana Luz P. Sangan. Today, we will present Pseudomonas species as a biological control agent. These bacteria play an important role in plant protection and sustainable agriculture. Throughout this presentation, we will explain what Pseudomonas are, how they work, and why they are important in pest and disease management. Pseudomonas species are gram-negative, rod-shaped bacteria commonly found in the soil, especially in the rhizosphere or the root zone of plants. They are known as effective biocontrol agent because they help suppress plant pathogen while supporting plant growth. They do this through several mechanisms. First is antibiosis, where they produce antibiotics that kills harmful microbes. Second is competition, meaning they outcompete pathogen for nutrients and space around the roots. They also release lytic enzymes that break down the cell walls of pathogen. Lastly, they triggered induced systemic resistance or ISR, which strengthens the plant's natural defense systems. Because of these abilities, Pseudomonas is widely studied and used in sustainable farming. To understand how Pseudomonas became recognized as a biocontrol agent, let's look at its history. In the 1970s, the term PGPR or plant growth promoting rhizobacteria was coined. During this time, researchers, especially at institutions like the University of California, began recognizing Pseudomonas as a powerful biocontrol agent. By the 1980s, research on Pseudomonas intensified. Scientists focused on how these bacteria colonize the rhizosphere and the antimicrobial compounds produced by different strains. Moving to the 1990s, the first commercial product based on Pseudomonas were registered with the USEPA. Examples include Blight-Ban, A506, and Biosave, which were used for controlling post-harvest diseases. From the 20s until now, research expanded to include genetic analysis. Scientists also defense mechanisms like induced systemic resistance, making Pseudomonas even more valuable in modern agriculture. And the rest will be discussed with my group mates.
Good day everyone, I am Anazavi Ibanez and today I will be discussing the uses of Pseudomonas sp and its target pest. First, Pseudomonas sp is primarily used as biocontrol agents or biopesticides against plant pathogens and pests promoting sustainable agriculture. So its target pests are fungal pathogens, nematodes or plant parasitic nematodes, insect pests, and bacterial pathogens. The first target pest is fungal pathogens. Under fungal pathogens are Rhizoctonia solani, Fusarium spp, and Phytium spp. Rhizoctonia solani, which causes dumping off and root rot. The mode of actions are antibiotics like DAPG, enzymes, and siderophores. Pseudomonas kills the fungus chemically, breaks it down, and steals its nutrients. Next is Fusarium spp, which causes welts and root rots. The mode of action is penicillin antibiotics and ISR, or induced systemic resistance. It weakens the fungus and strengthens the plants. Lastly, the Phytium spp, which causes damping off. The mode of actions are siderophore productions and volatile compound. It blocks the fungus nutrients and releases inhibitory gases. So next target pest is nematodes. Under nematodes, there are Melodogen spp and Globodera spp. Melodogen spp is a root knot nematodes. The mode of action is cyanide production, lipopeptides, and enzymes. The bacteria poison nematodes, which prevent egg hatching and degrade eggshells. Given in the picture is the Melodogen spp in Pacara airpod 3. Next is the Globodera spp, which is a potato cyst nematodes. The mode of action is DAPG, or diacetyl fluoroglycinol antibiotic, and protecyst. Pseudomonas prevent nematode larvae from infecting plant roots. Given in the picture is the Globodera spp in potato. Pseudomonas also suppresses some insects. Spodoptera litura, Spodoptera frugiperda, or armyworms, Plutelia xylostelia or diamondback moth, and leaf beetles and soil insects, or various insects that could harm the soil and your plants. Pseudomonas produces toxins and enzymes that reduce insect survival. It weakens insects through toxic compounds. And lastly, bacterial pathogens, Erwinia spp or soup rot bacteria, and Xanthomonas spp or leaf spot. The mode of action is antibiotics, which inhibit bacterial growth and compete for space and nutrients, so Pseudomonas blocks and kills disease-causing bacteria.
For the current trends, CRISPR-Cas9 o mediated genome editing for Pseudomonas fulva, a novel Pseudomonas species with clinical, animal, and plant associate isolates. These studies examine how modern genome editing, epigenetic inheritance, and dynamical systems modeling reveal emerging trends in bacterial genetics, germline programming, and cell fate control. CRISPR o Cas9 in Pseudomonas fulva, two plasmid red CRISPR system achieve 100% editing efficiency. Knockout of the 1.5 kilobytes and resistance gene impaired growth on chloramphenicol, confirming functional loss. Knock-in of a GFP cassette produced strong fluorescence, showing reliable insertion. Precise, high-efficiency editing is now feasible in non-model, clinically relevant Pseudomonas strains. This study explores how CRISPR or Cas9 technology can be used to edit the genome of Pseudomonas fulva, focusing on recent trends in genetic inheritance and dynamic system modeling. The research demonstrates high editing efficiency using a two plasmid system and confirms functionality through chloramphenicol resistance and GFP-associated fluorescent imaging. The study highlights potential for genetic applications and fundamental research in bacterial genetics.
Good day everyone, I'm Michaela Roloma. I'll be reporting the advantages and disadvantages of Pseudomonas sp as a biological control agent. Advantages. Pseudomonas offers several important advantages in agriculture. First, it provides strong protection against many soil-borne pathogens by naturally producing substances that inhibit harmful microbes. Because it is non-toxic, naturally occurring bacterium, it is also environmentally safe and serves as a healthier alternative to chemical pesticide. In addition, Pseudomonas improve plant growth by helping root absorb nutrient more effectively and by releasing growth-promoting compounds. It's also supporting soil health by increasing microbial diversity, which strengthen the overall soil ecosystem. With this benefit, farmer can reduce their reliance on chemical fertilizer and pesticide. Lastly, Pseudomonas also help crop tolerate stresses such as drought, salinity, and poor soil condition, making plant more resilient in challenging environments. Disadvantages. Despite these benefits, Pseudomonas also have some limitation. Its effectiveness can be influenced by soil conditions such as pH, moisture, temperature, and soil type, which means its performance may vary from one field to another. In some cases, the bacterial population decreases over time, requiring repeated application to maintain its beneficial effects. Another challenge is that certain strains can be difficult to store or transport because they do not remain stable in commercial formulation. Finally, when introduced into soil, Pseudomonas must compete with native microorganisms, and this competition may limit its ability to establish itself and function effectively.
Conclusion. Case strength. Pseudomonas sp offer eco-friendly, non-toxic protection against fungal pathogens, Rhizoctonia, Fusarium, Pathium, Nematodes, Meliodigene, Globigerina, insect, and bacteria, enhancing plant growth, nutrient uptake, and soil health while reducing chemical pesticide use. Mechanism and innovation. Effective antibiosis enzyme, siderophorase, ISR and toxins advanced by CRISPR-Cas9 for crisis genome editing in strains like Pseudomonas fulva, enabling high-efficiency modifications. Third is advantage. Promote sustainable agriculture, improve biotech stress tolerance, and boost microbial diversity. Challenges. Efficiency depends on soil conditions, requires repeated application, storage, formulation issue, and potential competition with native microbes. Overall outlook. A viable alternative to chemicals with ongoing research promising optimized strains for broader, more reliable agriculture integration.
That's all about Pseudomonas sp. Thank you for watching.